Welcome to my video on traceability of data in clinical trials.
Traceability of data refers to the ability to track and verify every piece of data throughout its life cycle, from its origin, through its transformations, to its final representation in analysis or reporting. In clinical trials, data traceability ensures that the data can be traced forward, from source to final results, and backward, from final results to its source. It establishes transparency, accuracy, and reliability, which are critical for regulatory compliance, audits, and maintaining data integrity. Importance of traceability in clinical trials. Ensures data integrity and prevents errors or fraud. Facilitates compliance with regulatory authorities, e.g., FDA, EMA, ICH, GCP. Enables auditors or reviewers to follow the data chain of custody to verify its accuracy. Supports reproducibility of results, a critical aspect of scientific research. How traceability of data is done in clinical trials. Data traceability is maintained through documentation, audit trails, and systematic processes. The following steps ensure traceability. 1. Data source identification. Clearly identify the source of all data points, e.g., CRFs, lab reports, electronic medical records. 2. Audit trail. Maintain a detailed log of any data modifications. Who, what, when, and why. 3. Linkage between systems. Ensure clear connections between raw data, transformed data, and outputs. For example, link EDC systems, electronic data capture, SAS datasets, and reports. 4th, documented processes, standard operating procedures, SOPs, and metadata for datasets are vital to map data flows and processes. 5th, data mapping. Data must be traceable across different formats, such as from raw data to SDTM, study data tabulation model, and further to ADAM, analysis data model, and outputs, e.g., tables, figures, listings. Real-world scenario, oncology clinical trial. Let us consider an oncology clinical trial investigating a new immunotherapy drug for lung cancer patients. Scenario details. Trial. Name, ONCO-123. Objective, evaluate overall survival, OS, and progression-free survival, PFS of the drug. Data collected, patient demographics, e.g., age, gender, cancer stage. Tumor assessments via imaging like CT scans. Lab reports, e.g., blood counts, biomarkers. Adverse events, AES, and vital signs. Survival status and dates of progression. Traceability process. First data source, raw data. Tumor size measurements are collected during CT scans and documented in the patient's medical record or source documents. Lab results, e.g., biomarker levels, are collected and stored in electronic lab systems, LIMS. Second, data entry into EDC system. Clinical site staff enter these raw data into the EDC, e.g., metadata rave, after verifying from source documents. Example, tumor size at baseline is recorded as 20 millimeters. Third, SDTM datasets. Standardized data. Tumor data is transferred into SDTM datasets following CDISC guidelines. ORS, observed result, shows the actual tumor size value, 20 millimeters. Fourth, ADAM datasets. Analysis-ready data. Data from SDTM are used to create ADAM datasets for analysis. Derived variables like tumor progression are calculated using RECIST 1.1 criteria. Example, if tumor size increases by 25%, progression is recorded. 5. Statistical analysis. Outputs. The analysis team runs SAS or R programs to calculate the number of patients with progression, time to progression, and Kaplan-Meier curves for survival. All programs include comments and input-output logs to ensure traceability. 6 Clinical Study Report, CSR. Outputs, e.g., tables and figures. In the CSR can be traced back to ADAM datasets, then SDTM datasets, and finally to the raw data. Example, investigating data traceability in a query. Suppose an auditor notices that in the CSR, a table reports tumor progression, 50 patients. The auditor wants to verify how this number was derived. Here's how traceability works. 1. Start with the ADAM dataset. Verify the para-MCD equals prog records for 50 patients. 2. Go back to the SDTM dataset. Confirm that tumor measurements, e.g., 25% increase, meet progression criteria. 3. Return to the EDC. Check if the tumor size entries align with the imaging source documents. 4. Source documents. Compare EDC entries with CT scan reports to ensure accuracy. The ability to trace data backward allows the auditor to validate that the final reported 50 patients with progression is accurate, reliable, and originates from the correct raw data. Key takeaways for oncology trials. Oncology trials rely heavily on imaging data, e.g., CT/MRI scans, and biomarkers. Traceability ensures tumor progression or survival results are scientifically sound and compliant with regulations. Tools like EDC systems, SDTM/ADAM standards, and audit trails make traceability achievable.
Many thanks for watching my video and be happy learning. Data traceability ensures your clinical data is audit ready. Please like, share, subscribe, and leave a feedback comment. Many thanks for your support and having a great connection.